Hello, thank you to Plexon for hosting this and for inviting us. My name is Ethan. I'm with the Morrison Lab in the University of Pittsburgh. And today I'll be talking to you about our work using optogenetic manipulation of dopamine neurons and how it can disrupt the acquisition of sign tracking and the cue reward encoding in the nucleus accumbens. Sorry. Okay, there we go. So first I should explain what sign tracking and goal tracking is. We use a Pavlovian conditioned approach task, which means that the animal is in an operant box and a cue will be randomly presented. Our cue is a light, tone, and a lever will appear out of the wall. This cue, which is the conditioned stimulus, would appear for 8 seconds. Afterwards, the unconditioned stimulus or reward is delivered. In our case, just a sugar pellet. And sign tracking behavior would be more interest in the cue rather than the reward delivery site, where goal tracking would be the opposite, where it's more interest in the goal delivery site or the sugar pellet. And we categorize them as sign trackers or goal trackers using a PCA index, which just stands for Pavlovian Conditioned Approach Index. And sign trackers are characterized as being above 0 on a PCA index, whereas goal trackers are below 0. And this is kind of an example of what a normal population would look like of sign trackers and goal trackers. As you can see, most kind of start out as goal trackers, and then the behavior is later acquired. And so why the interest in dopamine? Well, dopamine neurons, which are thought to signal for reward prediction error, are also very important for sign tracking behavior, shown by the Flagel et al. Paper here, that sign tracking behavior has a larger dopamine response to the conditioned stimulus or the cue. And actually, over time, the dopamine response to the unconditioned stimulus will diminish. And compare that to goal trackers. While there is some sort of dopaminergic response, it's about the same for both the conditioned and unconditioned stimulus. And so we wanted to ask, how can manipulation of dopamine neurons then actually affect sign tracking behavior? And so we decided to use a optogenetic manipulation approach. So we injected either channelrhodopsin or halorhodopsin into TH-Cre rats. And we would inject it in the VTA and implant optical fibers in the VTA. For channelrhodopsin, we did unilateral stimulation. And for halorhodopsin, it was bilateral inhibition of the VTA. And then we would wait 6 to 8 weeks after the virus injection and perform the optical fiber implant. And for a subset of animals, we also included an electrical or electrode implant in the nucleus accumbens core, as the accumbens is known for signaling for reward and reward valuation. And so we were interested in how signaling could change based on stimulation or inhibition. So for optogenic inhibition, we found that it would actually disrupt sign tracking acquisition. Here you can see the solid lines would be TH-Cre positive animals. So they would be affected by the optogenetic inhibition, and the dotted lines are just the wild type, so they're unaffected. And blue would represent sign tracking, while red or pink would represent the goal trackers. And we saw that during inhibition, which was the first 4 sessions, sign tracking was actually disrupted, the acquisition of the behavior. And once inhibition stopped, the sign trackers would then acquire that behavior. And here is an example of what a sign tracking behavior would look like, which is lever presses on the y-axis here. And it's kind of the same shape as the PCA index graph there. And here is an example of magazine entries, which would be goal tracking behavior, and that remains relatively unaffected by inhibition. Here is a look at what optogenic stimulation would do, which we found a little bit surprising to be disrupting subsequent sign tracking acquisition. So during the first 4 sessions, we would stimulate the VTA, and the TH-Cre positive and wild type rats were pretty much on the same trajectory behavior-wise until after stimulation, when further sign tracking acquisition kind of stalled out for the TH-Cre positive rats, while goal tracking was unaffected. And again, lever presses, kind of another way to look at what sign tracking behavior would be. And you can kind of see it almost plateaus there post-stimulation. And then magazine entries showing that goal tracking is unaffected. Now, for the optogenic stimulation of dopamine neurons, we found that it could actually enhance the nucleus accumbens reward response. So here is an example of an excitatory cell. You have on the left side with no stimulation, a kind of normal excitatory response to the reward. And with the stimulation, the VTA, there's a much more enhanced response to the reward. So you can see a higher excitatory response and it lasts longer during the stimulation. And here's an example of an inhibitory cell. So you have normal inhibitory response to the reward and then an enhanced inhibitory response. So just general kind of summary, we found that inhibiting the dopamine neurons at the time of reward will disrupt development of sign tracking behavior, but not goal tracking. And for stimulating the dopamine neurons, it will not actually change the acquisition of sign tracking, but post-stimulation, it would stall further acquisition. And for the stimulation, it did have a bit of a mixed effect on cue and reward activity, but it does seem to enhance responses to reward delivery. And a little caveat with this is that the behavior was already acquired when we see these effects, which is why right now we're actually looking into how stimulation and inhibition would affect signaling in the nucleus accumbens earlier on in acquisition. And thank you again for listening, to everyone in the Morrison lab for all of their help, and to Plexon.